Okay, today we are going to continue the next part that H type of polarographic cell we have to see. We have already seen the dropping mercury electrode and another type of the cell that has been used in case of the polarography is the H type of the polarographic cell. This polarographic cell diagram is in there, is present in your notes. You can see this diagram. In the diagram, there are the two compartments. One is the indicator electrode and another one is the reference electrode that has been joined with the sintered glass disc. Okay, in that there is a agar plug. That detail about this particular thing we will see. Even that indicator electrode will have the experimental solution, whereas the reference electrode should have the saturated KCl along with the calomel. Okay, this is the for the better understanding. I have shown this diagram. See here, this H type of cell designed by Legin and Latinen. In this particular type of the cell, the reference electrode such as saturated calomel electrode is placed in one arm of the dropping mercury electrode is inserted in the other arm, which also contains the experimental solution. The two arms are separated by cross membrane filled with the 4% agar salt KCl gel, which is held in place by porous sintered glass disc that I already shown. This particular part, you can see the bridge part is there. It is a agar plug. A darkened part is the agar plug that has been held by the porous sintered glass disc. Then the nitrogen can be bubbled through the experimental solution through the side inlet. The side inlet is there in this diagram. You can see from that particular side the nitrogen is going to insert. Then solution of this first rinse a number of times with the experimental solution and then filled with it. Then the DME capillary dropping mercury electrode capillary is then inserted into the experimental solution. H type of cell is connected in the polarograph setup in the same way as the DME mercury anode cell. Then the potential applied to the cell is varied and the corresponding currents are measured for the different potential. And the potential of the DME can be calculated as the difference between the EMF of the cell and the potential of the saturated calomel electrode, which is already known. This is about the polarographic H type of the polarographic cell. In the subjective type of the question, that question may come describe the H type of the polarographic cell used in the polarographic analysis. Okay, at that time you have to write about this point, you have to draw this diagram. Okay, then this video we will see.
Describe the polarographic technique using H-shaped polarographic cell. In 1922, T.Y.B.Sc discovered a technique with the help of which qualitative, quantitative, and even mixture can be analyzed qualitatively and quantitatively. The technique is known as polarography. In this case, electrolysis of experimental solution is done. The cathode is polarized electrode, where deposition takes place, and anode is non-polarized electrode. The cathode is dropping mercury electrode, and anode is large pool of mercury or reference electrode, such as saturated calomel electrode with large surface area. H-shaped polarographic cell consists of two electrodes in two parts. One is indicator electrode or dropping mercury electrode, where reduction takes place, and other is reference electrode like saturated calomel electrode. Both half cells are connected with the help of agar plug. Experimental solution is filled in chamber of dropping mercury electrode. Nitrogen gas is passed for about 10 to 20 minutes to remove dissolved oxygen from experimental solution. Saturated calomel electrode or anode is connected to the variable resistance and then to positive terminal of battery. A sensitive current recorder is connected in series and a voltmeter is connected between cathode and anode. The height of mercury reservoir is adjusted that mercury falls at the rate of 20 to 30 drops per minute. Close the circuit and keep the jockey S on X position. For this position of jockey, find out the reading of voltmeter and galvanometer. The jockey was moved from X to Y slowly, and for each position of jockey, take the readings of voltmeter and galvanometer. Now, plot a graph of current versus applied voltage. The graph thus obtained is known as polarogram. As half-wave potential is characteristic of each ions present in solution, we can have qualitative analysis, and with the help of the diffusion current, we can have quantitative analysis. D. Elkovic performed the number of experiments on polarography. In each case, he observed the applied potential and current obtained in galvanometer. In each case, he plotted graph of current versus applied potential, and from graph, he calculated diffusion current ID. He said if ID is known to us, we can find out the concentration of experimental solution. By equation, ID is equals to 60C N D power to half M power to 2 by 3 T power to 1 by 6 and C, where N is valency of metal, D is diffusion coefficient which is a constant, M is rate of fall of mercury in grams per second, C is concentration in millimoles per dm cube, T is the average time of fall of mercury drop, and ID is diffusion current in microamps. As D depends on temperature, therefore ID also depends on temperature. And if you like this video, please
Okay, thank you for this today's lecture. We'll stop here.